nanoHUB-U, online instruction.
Welcome back. I'm Professor Rickus, and we're going to talk about protein half-lives in this lecture. We're going to dig a little bit deeper into the protein half-lives that we started talking about previously, and we're going to look at the gene product decay rate constant alpha from our previous Hill function models of gene expression. And we're going to look at what does the half-life of real proteins look like, and does this model hold up in real systems? So let's return to our basic rate equation for gene expression, where the rate of change of level of protein Y is just a function of generation, expression of our gene, and consumption. And that consumption can be either degradation, passive or active, more often active. Proteins are very actively and tightly regulated and controlled through degradation in most of the time in cells. Or we could also have decay of our protein level via dilution just through cell division. So an overall effect of on our rate decay constant is going to be the sum of the dilution and degradation decay of our protein. So in the case of degradation, alpha degradation is going to be very small for a very stable protein, whereas alpha degradation would be large for a rapidly degraded protein. And for looking at alpha dilution is going to be large for very rapidly dividing cells, such as E. coli, and alpha dilution would be very large for very slowly dividing cells. So again, let's look at our two extremes. I just mentioned one. If we considered rapidly dividing cells, such as E. coli in culture in the lab, we would see that dilution is really going to dominate that decay rate because it's so rapid relative to the rate of protein degradation. Whereas in very slowly dividing cells, or the extreme of that, a post-mitotic cell such as a neuron that's not dividing, the protein half-lives are going to be dominated by degradation. And we can often then approximate our decay rate by the decay rate of degradation of the protein, because it will be dominating in these slowly dividing cells. So we can make some testable predictions from our system. So again, back to our basic rate equation. If we could eliminate or prevent the production of protein, so this beta fx term would go away, then our rate equation would just collapse to a simple exponential decay. And our model would predict then, if we measured levels of protein Y over time, and we fit the natural log of that protein level as a function of time, we should be able to fit our data to align with a slope that's determined or equal to that overall decay constant alpha. So here we have a testable prediction. And so what you can do, and what has been done in this paper here, is that this model was tested in real cells and said, if we go in, do we in fact see this type of behavior that our model would predict? So for the rest of most of this lecture, we're going to be digging into some of the details of this paper that was published in Science in 2011. And so first, you can ask the question, how do you measure protein half-lives in real living cells? Well, one way to do this is through this Bleach chase experiment that they devised. So what they've done here is they've taken lung cancer cells, and they have an array of these cells, each that has a different protein that's been fused to a fluorescent tag, in this case YFP. So now we have an array where we can watch all these different cells that are expressing a particular protein, and we can measure that protein level by fluorescence over time. So here we've got, you can get protein level over time, and they could do this in individual cells. So they have a whole bunch of cells here by these small gray lines, and you can look at the average by this thick black line. So, okay, fine. So there you can follow the protein levels. But as we know, protein levels are going to be affected by production and decay. So how do we experimentally get rid of that production? So they came up with a clever trick using a Bleach chase workflow. And so what they do here, the idea is, in this top row, you have all the proteins that are labeled with the fusion YFP, right? So they're all going to be glowing. We can see them when we excite them. So then what they did was at a certain time, we'll call time 0, they bleached. So you can bleach the fluorophore, and some, perhaps maybe not complete bleaching, but some proportion. So we see we have a couple proteins left that might still be fluorescent, and we have a bunch now that have been bleached and are now invisible to our microscope. So what will happen as the cell produces more, that population of fluorescent will begin to grow. But what's important here is that any new protein will be fluorescently labeled. So now what you can do, we can't measure the invisible protein directly, but we can indirectly via a subtraction. So if you take the total fluorescent protein over time, and you now subtract the visible protein, and remember the new visible protein can only come from production, and you subtract those, and what you're left with is the population of invisible protein that became invisible at that time of bleaching. So what they could do then was watch the decay of this population over time, because we can't make more invisible protein. Any of the new protein is going to have the fluorescent tag. And so then could take that and plot now the level of the invisible protein that comes from the subtraction of these two measurements and plot that value versus time. And we should be able to follow an isolated decay or removal that should be a single exponential decay if our model is correct. And so what would we expect from the curves? So here we have some theoretical curves that they show us in the paper. And so the idea here is that at time 0, what we've got here is a curve of our blue. That would just be our normal total protein production. And if at time 0 we bleach, so now you've got this gap, right? And that represents now that we've got some small population that's still visible, but this gap represents that invisible or bleached population. So if that population were rapidly removed, if it rapidly decayed, we would expect that difference to decay very rapidly to eventually being no difference. If we had a medium level of removal, then we would see this gap decay, but more slowly. And if there were no removal of the protein, no decay of the protein, our invisible population again, you would see that gap maintained through the time course of these curves, right? And so then that differential you can plot as a function of time, and what you would expect that again for a very large alpha decay rate, we should see a linear plot when we plot the natural log versus time of that invisible population, and the slope of that line will be alpha, and it should be steep. For a medium decay rate, right, we should see a similar curve, but the alpha, the slope, is going to be less severe. And in the case, if there's no decay of the protein, of our invisible population, then alpha would be 0, and we should see a straight line. And so the question is, what did they see? And they were, in fact, able to fit these exponential decays to an array of proteins. They did about 100. And so then what did the half-life, what did the alpha decays of these real proteins, these 100 proteins, and the corresponding half-life of those proteins look like? So here's their histogram. And first in the top, we've got the decay rate, our alpha. And remember, alpha is our protein removal rate that is made up of both dilution and degradation. We have in units inverse time here. And they found that alpha dilution, so remember, alpha dilution is going to be determined by the doubling time or the growth rate of the cells. And for these particular lung cancer cells, gives an alpha dilution of about 0.03, plus or minus here 0.004 inverse hours, okay? And so you see this histogram here on average of these proteins. They saw about 0.03 inverse, or range of 0.03, which is extreme from the dilution rate, to 0.82 inverse hours with an average of about 0.1. Okay, so it's maybe a little bit harder to think about the decay rate. So let's flip that around and think about half-lives now and in units of hours. So just to remind you, our half-life then is going to just be the natural log of 2 over our decay rate. And so if we look at this now in hours, something I think we can a little bit easier wrap our brain around, we can see the extreme there is that cell cycle of the cell, because when the cell divides, it's going to cut the population in half. So that sets sort of our maximum extreme. And within here, what they saw was a wide range of half-lives between 45 minutes and the doubling time of the cells, which was 22.5 hours, with a mean of about 9 hours. Okay? So we can make further predictions. So what we know so far is that they were able to isolate exponential decays of a population of cell, and they were able to fit that data and then measure directly the half-life of those proteins inside individual cells. So we can expand our model a little further and make some additional predictions. So here's what they did. They asked the question, well, what if you add a drug? What if you add a drug that might change either? You could look at the effect of that and think of, in one extreme, if the drug only affected degradation, then we can add this term here that represents our drug, K1. So if our drug were to have the effect of reducing the degradation of proteins, then we would expect this K1 then to be in a value between 0 and 1 and no change in our alpha dilution. So we can then convert that and look at our effect of our half-life, our new half-life after drug treatment, which is indicated by star, compared to the half-life for each protein before drug treatment. On the other side, if the drug were to reduce dilution, in other words, to affect the growth rate of the cell, we can create a similar model where here they put in now a K2 term that would be between 0 and 1 that would reflect some reduction in alpha dilution, right? So that's going to bring that term down by some percentage determined by our K2. So then the model makes predictions. So here's an example. If you looked at all 100 proteins and you plot the new half-life of that protein with the drug versus the half-life of the protein without the drug, right? If there were no effect, if they were equal, we would get this straight line with a slope of 1, right? So your model then would predict, we'll put in here, they put in a K1 of 10%, right? So there's a 10% change, that your new alpha degradation is 10% with the drug of what it was without the drug. And you can predict what the new half-lives of the protein would be. And you can see this upward curvature type prediction that you would get if our drug had an effect on degradation. Similarly, you can do the same thing and say, what would be the predicted new half-life after drug treatment if our drug affected the dilution, that is the growth rate of the cell. And again, here, 10% of the original value with drug. And now we can see that we see a different shape in this curve. So we have quantitative and qualitative predictions. And now what you can go do is actually go in and treat with different drugs and see how well this model holds up to predicting effects on degradation and dilution. And so this is what they did. So, and let's take an example of one of the drugs they looked at. So they applied an anti-cancer drug, which is known to arrest cell growth. And let's just look at 2 examples of the proteins that they of the 100 that they looked at, right? So here's DDX5 and DDX5 during normal growth conditions and after drug treatment. Okay. And so what they see here then is you see a decrease in the decay rate alpha. And they saw this global decrease in the protein half-lives of a wide range of proteins that they looked at. In 2 examples, you can see this in the DDX5, the alpha is decreased. For another protein example here, the LMNA is also decreased. So, and then the question from this, you can't tell just by looking at alpha what the contribution from dilution and degradation is. And so we do know that CPT arrests cell division. And so the question is, if we put that into our model, does our experimental data fit our model? And this is what they did. So they put in, because CPT is an extreme and fully arrests cell growth, they put in a K2 value of 0, right? So there's no dilution term. And fit what you have here, the red dots are their actual data, and the blue line is the fit from the model. And as you can see, both qualitatively and quantitatively, the model was able to predict and have a good fit to the experimental data across the proteins that they looked at. And so again, you can see for these, and there's a difference in, so if we look at what the predicted is and the measured is after drug treatment versus the initial half-life of the protein, we get a good fit to the experimental model. Experimental data gives a good fit to the model, excuse me. And this holds up quantitatively and quantitatively as we look across a range of different K2 values. And so they looked at this experimentally. They applied drugs that altered the cell doubling time to varying degrees. Right? So the first drug that we talked about previously was extreme and completely arrested cell growth. There are other drugs that will affect, and treatments that will affect cell growth, but to different extremes. So they looked at, another drug that completely arrested cell growth. They looked at starvation, which, had, an arrest of cell growth pretty large, but not quite as extreme. And other K2 values ranging from 0 to 85%. And the point here is that the design curves here could really predict the effects of altering the cell cycle time on the protein half-lives. So they saw a good fit by putting in the change in their dilution rate from the doubling time of the cells, and were able to predict the change in half-life for all the proteins that they were looking at. Okay, so let's think about some of the functional consequences of this outcome. So perhaps we've sort of convinced ourselves that the model provides a good fit and is reflecting what happens in real cells, in this case, these lung cancer cells. But let's think a little bit more broadly about the functional consequence of this. And so the first is that the proteome of more rapidly dividing cells will get, and by proteome I mean all the proteins that are being expressed in the cell, the proteome will get more out of whack in rapidly dividing cells when growth is arrested. So if we look here at this plot, so if you think about in a range of very rapidly dividing cells from 4 hours to a day, compared to slowly dividing cells that may have a doubling time in the order of a week to no division. And we look now at how these curves would change if you were to totally arrest division with a drug, and plot again the predicted half-life with the drug or treatment compared to the initial. So for cells that have no division, obviously if we arrest their growth, that's going to have no effect, right? Because they're already not dividing. And so this remains a 1-to-1 correlation. There's no change in the half-life of the protein. But if you were to arrest growth, depending on that initial growth rate of the cells, you can see much more extreme changes. So if you look at a difference down here for some of these proteins, right, that some would go to very long half-lives that were initially very small. So that difference is very large, and the entire proteome, looking across all this range of half-lives, is going to get much more out of whack when growth is arrested. Okay. The other consequence here is that long half-life proteins are more affected by growth arrest than short half-life proteins. So if you look in these curves, so again, our half-life after treatment or in our new condition versus half-life in the original, if we look out here at longer half-lives, before and after, they deviate further from this 1-to-1 line, right? So the proteins that have initially naturally stable long half-life proteins are going to get much more out of whack relative to the shorter half-life proteins when you growth arrest. And so this may have very important consequences when we're engineering systems. Perhaps we're immobilizing cells into a bioreactor that might slow down or change their growth rate. And how might this change the proteome and the expression and dynamics of gene expression in those cells? So there's lots of consequences of this if we begin to think about some of the engineering applications in which we use cells, as well as, such as in this context, drug treatment for treating different cell ailments, or in this case, the cancer example that we looked at. So let's look at some other implications of the model by going back and looking at their data in a little bit different way of all the proteins they looked at. And so here they sorted the proteins by short half-life and long half-life proteins. And it's useful to sort of look at the relative, which ones were really dominated by degradation, which ones were really dominated by dilution. So about 48% of the proteins were really dominated by degradation under standard culture conditions. About 42% really, you couldn't say were dominated solely by one dilution or degradation. They really were influenced, the half-life of the proteins were influenced by both of those. And about 10% of the proteins were most dominantly affected, their half-lives were most dominantly affected by dilution. Okay, so again, this is that concept that we talked about before, thinking that degradation dominates short half-life proteins, in this case about 48% of the 100 proteins that they looked at. Dilution dominates half-life of the proteins. Again, about 10% was the case in the 100 proteins they looked at. And the other interesting thing that came out of their analysis was that, to some extent, proteins with a common functional cluster tended to have similar half-lives. So those that were involved in translation and initiation, for example, tended to cluster down here in this mixed and dilution dominance, whereas those proteins involved in cell cycle tended to be cluster more down in this degradation dominance and mixed dominance. And so that has some interesting functional consequences for our systems as well. Okay, coming up, we're going to start moving into some gene circuit motifs, starting with autoregulation, and importantly, starting to look at some of the design features that are enabled by these motifs, such as robustness.